Thank you. So I'm going to talk about a few different aspects of this problem of phagocytosis, which is crucially important in our immune systems, both for fighting bugs, but also for clearing dead cells. Oh, I should acknowledge people first of all, just in case I forget to acknowledge them at the end. So these are some of the faces of people who've been working on different aspects of this problem. I realized I forgot someone. Antoine should be there as well. Sorry, he's not there. Okay. So to introduce this, I want to show you my favorite video of all time. This is a white blood cell chasing a bacterium. The round things are red blood cells. You can ignore them for the time being. The great big white thing in the middle is chasing the little bacterium that's floating around. And I need to unmute myself maybe. Okay, for the Zoom. Right. Okay. Yeah. So, I didn't talk through the exciting bit. I hope you were watching. I can run that again. So eventually what happens is this white blood cell engulfs the bacterium eventually. And if all goes well, it will then kill it. And the immune system has got rid of this nasty bug. So it eventually gets there. There we go. And it goes inside. Okay, so this
chasing her, or is it pushing it away? Notice it's always about the same distance.
Okay, so there's a lot going on here. So there's all sorts of questions to do with chemotaxis. Is it chasing it? Is it following chemical gradients? I'm not going to be talking about that aspect of it today. There's obviously the cell migration aspect of this, but those of you who know me know that that's been an interest of mine for many years, using some of the theory that Carsten was talking about earlier to look at models of cell migration. What I'm going to be talking about today.
There's also low Reynolds number.
Yes.
It's anyway.
Yes, there's lots in this video, which is why it's my favorite video. It's the one that has inspired my entire career, basically, and probably will continue to do so for the rest of my career. But what I'm going to be focusing on today is the process of eating the bug, which is called phagocytosis. So this is an electron micrograph showing in quite nice detail a cell just in the process of starting to eat a bug. And the nice thing about this in terms of experiments is you can play around with feeding these things beads. They will eat latex beads, and you can give it beads of different sizes and play around with it in that sense. So I'm going to be showing you a variety of real bugs and beads. The video you saw before was a bacterium. I'll be actually focusing on single-celled fungus later on because I've got a biologist collaborator who's interested in that in particular. I like it because it's spherical, but there's all sorts of interesting things that happen to it as well. So just an outline, I'm going to give you an introduction. I'm going to be talking about actin polymerization. Actin has been an interest of mine throughout my